Hi everyone, I'm Meenakshi and I'll be presenting our work on a shared neural encoding model for the prediction of subject-specific fMRI response. This is joint work with Gia, Keith, Amy, and my advisor Mert R. So let me start with a basic description of what encoding models are and how they can be useful in neuroscience. Encoding models are popular computational models in sensory neuroscience that are used to predict how neurons or populations of neurons across the brain respond to complex naturalistic stimuli. These encoding studies typically use machine learning methods to predict fine-grained brain activity from stimulus features. At this point, it is important to ask what is the use of these encoding or predictive models in cognitive neuroimaging. In the past, sensory systems have been studied extensively using task-based paradigms, where the brain activity is recorded upon stimulation with handcrafted stimuli. This paradigm has been very successful, for example, in identifying scene-selective or face-selective regions in the brain. While successful for testing specific hypotheses, this approach is limited in the sense that no single task-based experiment can help in developing broad theories of sensory processing in the brain. Predictive models, on the other hand, are based on out-of-sample prediction, and they generalize to arbitrary new stimuli and can thus offer more holistic descriptions of sensory processing in an individual. The biggest advantage is that once we have such a general model, we can use it to generate novel hypotheses about information processing that can then be tested under more controlled conditions. These models can also be used in other applications, such as neural population control, by optimally synthesizing stimuli to elicit a desired neural activation pattern. As a main motivation behind our work on shared neural encoding, we know that building accurate subject-level models of brain function often requires large amounts of data per subject for good generalization. However, most encoding studies are constrained by the size of their stimulus set due to the time-consuming and expensive data acquisition process. Further, subject-level data, especially if acquired in single trials, can be very noisy. There is very limited work in leveraging multi-subject data for more robust and accurate individualized neural encoding. In this study, we attempt to address this limitation, and our main goal is to leverage group-level data to improve subject-specific response prediction. In the present study, we develop both visual and auditory encoding models that predict fine-grained voxel-level brain activity patterns from raw images and auditory spectrogram inputs respectively. These neural encoding models comprise two components: a feature extractor which pulls out the relevant features from raw images or audio waveforms as shown by the S vector in the figure, and a response model which maps these stimuli features into brain responses. Ideally, we would expect inter-subject knowledge transfer to be successful if different subjects share similar cortical representations of naturalistic stimuli. Indeed, recent studies have revealed that coarse-grained response topographies are highly similar across subjects, and naturalistic stimuli such as movies engage a large portion of the brain, invoking a response that is shared across individuals. In light of these observations, we propose the illustrated shared encoding framework with a novel convolutional response model. Assuming that individual differences mostly occur at a finer scale and that there is a significant response component that is shared across individuals, we propose to leverage a shared response model such that early layers of this model, which capture coarse-grained responses, are common across subjects, while the deeper layers, which predict the fine-grained responses, are subject-specific. This is similar to a multitask learning setup, where we are using a single shared architecture to predict the responses of multiple subjects. The main advantages here are that we can combine data from multiple subjects into a shared model, and this approach allows simultaneous optimization of encoding models across subjects. Further, since only a handful of convolutional layers are subject-specific, it poses minimal memory overhead with additional subjects, and can thus handle fMRI datasets with a large cohort. Finally, this approach is amenable to incremental learning with diverse varying stimuli across subjects, with less constraints on data collection from single subjects. In our experiments, we study 70 fMRI recordings from a randomly selected sample of 10 subjects from the HCP movie watching database. The dataset comprises 4 audiovisual movies, each nearly 15 minutes long. We split the first 3 movies for training and validating our models, and use the fourth held out movie for independent testing of our models. We compute log-mel spectrograms over every 1 second of audio waveform in the movies to obtain a 2D image-like input for the audio feature extractor. and extract the last frame of every second of the video to present to the visual representation network for obtaining visual features. Here is a short glimpse of the stimulus and response data for a single movie in the HCP dataset. The left figure shows the average response across all subjects, whereas the right figure shows the stimulus frames. The subjects also listened to the corresponding audio while lying inside the scanner. We evaluated all our models by computing the Pearson's correlation coefficient between the predicted and measured response at every voxel, and summarized the performance of models by computing mean correlation across all stimulus-driven voxels. We compared our approach against encoding models with the same architecture that are trained with single subject data, as well as against individual level encoding models that employ a linear response model instead of the proposed convolutional response model. The top row shows the performance of all auditory encoding models, whereas the bottom row shows the performance for visual models. In both cases, we find that the shared encoding model consistently performs better than individual encoding models that are trained separately on single subjects in terms of both the mean correlation as well as the number of voxels that show significant correlation between the predicted and measured response values. Finally, it is important to note that the shared model does not simply predict a mean response, but indeed captures meaningful individual level idiosyncrasies pretty well. This is most clearly seen by the diagonal nature of matrices on the right hand side, which show that the predicted response for a subject best matches the actual response of the same subject. We also visualize the correlations between the predicted and measured fMRI response across the cortical surface for both the proposed models. The figure here shows the average correlations across all the 10 subjects in our study. For the auditory model, we see significant correlations in the parabolic auditory cortex, extending into some other language areas as well. For the visual model, while we see significant correlations across the entire visual cortex, the performance is much better in higher order visual regions across both the dorsal and ventral visual stream. Next, we wanted to investigate the possibility of using encoding models as neural activity synthesizers. Given that we have a predictive model linking stimulus and brain activity of each subject, we can feed it stimuli from alternate paradigms such as task fMRI. Here, for example, we used face and scene stimuli from the HCP working memory task. Next, we used synthesized or predicted neural activity for these stimuli to generate subject-specific contrasts and computed the dice overlap between the predicted contrast for each subject against the true contrast of every subject to produce an n cross n matrix for each contrast, where n is the number of subjects. The diagonal dominance in the dice matrix for both face and scene contrasts suggests that the predicted contrasts for a subject are most similar to the same subject's true contrast. Further, we didn't observe a prominent diagonal structure for individual subject models, presumably because they generalize poorly to out-of-domain stimuli. Importantly, the predicted contrasts highlight areas previously known to be involved in the processing of faces and scenes. For example, the fusiform face area is consistently seen in face contrasts, whereas the parahippocampal areas are seen in the scene contrasts. This agreement with results known from task-based experiments suggests that these encoding models could indeed have applications in personalized brain mapping. To conclude, we've made a promising step towards shared neural encoding models. This approach enables us to build predictive models of neural responses across different subjects in concert rather than in isolation, and thereby facilitates inter-subject knowledge transfer. Further, it allows us to exploit the redundancies or shared response across subjects so that we can build much better predictive models of neural responses with limited data from individual subjects. To enable end-to-end optimization of the shared model for whole brain response prediction, we proposed a convolutional response model that dramatically reduces the number of free parameters in comparison to a linear response model while yielding better predictions. Finally, we found that these models can generalize remarkably well to out-of-domain stimulus sets from alternate cognitive paradigms, and thus demonstrated their application as virtual neural activity synthesizers for personalized brain mapping. Thank you for listening. I'd be happy to take any questions during the oral or poster session.